Today we will be talking about gene expression databases. Gene expression databases. So, to understand or to study gene expression databases, we must first understand what are gene expression processes and methods, meaning what type of data is generated in gene expression studies. And then we will talk about the databases, that how this information is organized in databases and how we can retrieve this information from there and use it for our purposes. And we will take up the example database as NCBI Geo. So, in the section 1 we will talk about the methods and processes and the type of information which is obtained in these studies, and in the section 2 we will talk about how this information is stored. So, starting with the this section, in this section we try to answer 3 questions. The first is, what is gene expression and how it is different from global gene expression. We will talk about gene expression, that why global gene expression is very important for genome sequencing projects, and what are the different methods which are employed in gene expression studies, and in a way what sort of data it generates in the studies. So, we begin with global gene expression. What is global gene expression? Before global gene expression, we know already that gene expression is the process by which information from a gene is used in the synthesis of the functional gene product, or maybe a protein, which in usual cases is a protein, but we have few non-protein coding genes also, and they only code for RNAs, maybe transfer RNA or small nuclear RNAs. So, the genes may be protein coding genes or non-coding genes, and the genes typically are located on chromosomes. The genes basically may not be the continuous expressing sequence, rather they have intervening regions, those we call as introns. So, genes are made up of exons and introns both, and how they express themselves, meaning how information flow from gene to a product, to its product or protein maybe. This process is maybe a 2 or 3 step process. The first step here is called as transcription, where mRNA is synthesized from the DNA in the gene, which has both exons and introns. So, the synthesis of mRNA from DNA is called as transcription, and the introns also are transcribed. After this, we have RNA processing or splicing, where the intron region is spliced off and we have a product synthesized that we call as mRNA. All this takes place in the nucleus, and after the mRNA is ready for the other process, that is the translation, in which the information encoded on this mRNA is used for synthesizing a amino acid sequence, that is a polypeptide. That process is called as translation, and for this process the mRNA comes out of the nucleus in the cytoplasm and does the translation and synthesize a polypeptide. So, this is the basic flow of information, and this information is contained in genes, but there are certain important things which makes it not so simple process, like not all genes are expressed in all cells at the same time. Gene expression depends on cell or cell type or tissue type. Sometimes the stage of the organism developed decides that which genes are to be expressed. We have health or disease conditions, and the environment also controls that which genes are to be expressed. So, that way we are trying to say that there is a regulation on expression of these genes, and that is a very important process. And for understanding this regulation of gene expression, If we study single gene expression, it's very hard to retrieve any information when we study single gene expression. So we can study gene expression by taking up a single gene, or maybe we can study the gene expression as a global gene expression. What we do in global gene expression is that we study the quantitative relative expression of many genes at one go, and we estimate, or we know the expression of population of RNAs in the cell. So we study many genes and we talk about the quantitative relative expression. This is what we call as global gene expression. So in global gene expression, we study the expression of many genes and we talk about their quantitative relative expression as population of RNAs in the cell. So we can know the variation in gene expression, and we can also have an idea of what is controlling in what situation. So global gene expression is a very powerful tool in molecular biology, and it is used to explore basic biology and to diagnose diseases. It facilitates drug discovery and develop therapeutic to specific pathologies. So it is an extremely powerful technique, and it reveals several important and astonishing facts from the genome data. Basically, we want to study gene expression, meaning in the genomes we get a lot of information about the relations and regulation among the genes while studying gene expression. But generally, when we plan to study gene expression in the gene expression databases, we may be trying to understand the basic biology, or we may be understanding the genetic variation, that is, what change in a gene is leading to the change in the regulatory elements, or maybe the phenotype controlled by their gene, or maybe we are trying to understand the factors that makes gene expression go wrong. And we might also like to address and treatment and prevention of a disease by studying gene expression. And we might also be intending to apply this in biotech industry, that is, maybe we are studying genetically modified organisms and we are studying the expression of genes there. So studying gene expression may be serving several purposes, but here we'll be talking more about their role in genomes and how their interrelation is making it's very important, and that becomes inevitable to be used without bioinformatic resources. So we now are moving on to the next question of ours, that is how global gene expression is mainstay in genome sequencing. The gene expression, single gene expression, could have been studied without bioinformatic resources, but the global gene expression is very, very important in genome sequencing projects and in getting information from the genomes, and that cannot be done with bioinformatic resources. So first we study how global gene expression is important in genome sequencing. So what we do in genome sequencing projects, our main goal is to understand the gene expression, not only genes but all the genes together. So how we do this is we first have genomic sequencing done, and then we identify these sequences, and then we determine the all set of genes present there. After that, what we're going to do by knowing the genes, which gene is located where, our final goal or the ultimate goal is to understand when, where and how a gene is turned off and when it is turned on. That is how this gene expression is controlled or how it is regulated in a cell. So in a genome, gene expression is regulated through complex mechanisms. Like we have numerous gene products that are involved. Those may be proteins or regulatory RNAs. They may be interacting among each other through numerous pathways or regulatory networks, or they may be interacting with the genome, and that in turn makes it a very complex process and requires sophisticated tools to study this. And genomic gene expression studies are different, meaning you require special skill and tactics to study that, because the genomic DNA encodes for numerous functional elements, and the many genes that do not code for proteins, rather they code for these regulatory RNAs, and these regulatory RNAs sometimes they control the gene expression, and at other times there are other factors like DNA methylation, chromatin structure, histone modification. So rather than gene sequence, there are other heritable changes in the gene expression. Those also need to be taken into consideration when we are studying gene expression in genomes, or we are studying many genes expressed at one go. So this cannot be done when we study one gene, one enzyme. We need to have global gene expression, and we must study hundreds of the genes at one go in a genome to visualize or to solve the questions of gene expression while studying the genome data. So with this one, we conclude that global gene expression studies are very, very important. The genome gene expression cannot be studied without the global gene expression, and that in turn depends upon the bioinformatic resources, the databases and the softwares. So moving on to the next question which we were trying to understand, that what are the methods in gene expression studies, meaning what sort of data would be coming out of these gene expression studies, so that we need to organize that in databases, and we should facilitate their retrieval while studying gene expression in genomes. So the methods for the study of gene expression have evolved from maybe after the discovery of DNA in 1953, after the central dogma was given. It moved from several ways, but now several steps, but now we have next generation sequencing methods. Those are very rapid, and we also have EST, so microarray methods also. We have moved on from one method to another, but here we will be talking about only EST and DNA microarray methods, because those are the methods which yield the data which needs to be stored as databases, and which can be used by other scientists as it is, and can be related to, analyzed to infer, to reveal the hidden facts in these data. So this is just a small picture which is showing that we can study gene expression by a single gene, but that would be simply studying a mRNA sequence in GenBank. But we can study batch gene expression, meaning many genes together, and for studying that we do what? We may sometimes start it with mRNA, because mRNA, as we studied in the previous, observed in the previous diagram, the mRNA which is processed from pre-mRNA by removal of the intron, it is the final product. The introns are removed from there, and that can be used to study the gene expression, because that is the final product of expression. But what happens in this mRNA is that this mRNA do not contain certain sequences from the regions, meaning it is good that the introns have been removed, but the problem is that it is available in the cell for very small time, meaning it is unstable outside a cell. So therefore what happens is that we needed to design other methods to understand the sequence or the information contained in this mRNA. So with the help of the special enzymes like reverse transcriptase, scientists designed methods to synthesize complementary DNA, that is very commonly called as cDNA, from this mRNA. So what is done is, with the help of mRNA, with the help of enzyme reverse transcriptase, complementary cDNA is synthesized from this mRNA, and the benefit of this cDNA is that it is much more stable compound, and because it is generated from the mRNA, so introns are removed. So this shows you the final product, meaning the final sequence which would be representing the product. So this cDNA is advantageous, and we could use this to synthesize a double stranded cDNA. And what happens is that this cDNA for study of gene expression are important in one way also, because they contain the ESTs. What is EST? EST is expressed sequence tags. So expressed sequence tag is a tiny portion of an entire gene that can be used to help identify unknown genes and to map their position within a genome. So what are ESTs? They are small pieces of DNA sequences, usually 200 to 500, that are generated by sequencing either one or both ends of an expressed gene. So the idea is to sequence bits of DNA that represent genes expressed in certain cells, tissues or organs from different organisms, and use these as tags to identify the genes. So the beginning portion, meaning this EST have the cDNA has 5' end and 3' end. So EST present at 5' end is called as EST, 5' EST. A 5' EST is obtained from the portion of a transcript that usually codes for a protein. These regions tend to be conserved across species and do not change much within a gene family. So 5' EST is a conserved region, and that usually remains conserved across the gene families. Similarly, we have 3' EST, and these 3' ESTs are not conserved. They usually fall within the known coding or untranslated regions, and they are usually not conserved across the cross species. So both these ESTs, let's review this again with the help of this diagram about these ESTs, is that we said mRNA is relatively unstable structure. So to work further with this mRNA, what we do is we synthesize a complementary DNA that we call as cDNA. Now this cDNA is done what? At both the ends of this we have two types of ESTs. One is 5' EST and the other one is 3' ESTs, and both these EST sequences, they can be used to tag or to identify a gene, and they can also be used to identify their location on a chromosome, on a genome. So ESTs have been very useful in gene identification and their location on the chromosome. The EST method was used for a very long time, and what has happened is they are small sequences, and what has happened is because of this next generation sequencing data and the new emerging sequencing technology, the focus is shifting in favor of next generation data, which is directly deposited to SRA, the sequence read archive. So people are, meaning according to the studied data, people are using much less EST data. So because of that, the NCBI, it merged its EST data with the GenBank data, and now they do not lie as exclusive data, and the main reason for doing this is the availability of next generation, availability of, I would say, short reads generated by high throughput sequencing, the gene expression in genomic data. So we study many genes at one go, and if we look at DNA microarray is like a glass slide which we observe under microscope, and what it does is it has grooves, many grooves to which a DNA chip, this is also called as DNA chip, and in these grooves the oligonucleotide or pre-read oligonucleotides referring to genome data are attached. So it's a simple glass slide and it has the genomic DNA attached to this. The pre-sequenced already known gene data is attached to this, and it is extensively used in measuring gene expression, and with this we can study large number of genes at one go and study their expression together. So microarrays are what? They are the simple glass slide or silicon slides upon which DNA is attached as an organized or predetermined grid fashion. So each spot of DNA is called as a probe, and that represents a gene, and that represents a gene. And what I would like to mention here is that we need to have a proper organization of this data, meaning the groups, when we have thousands of the groups on this slide and each of them contains a gene or an oligonucleotide, before performing an experiment we must have a proper information about what is the probe, meaning the DNA microarray chip which we have used has probes and which genes are loaded on these probes. And I said we can analyze tens and thousands of genes simultaneously with DNA microarray. Then let's understand what happens and how we study this gene expression with DNA microarrays. So what we normally do in DNA microarrays is not that we load only one sample, we load many samples and we have standards also. So meaning the one side of the array maybe we have different genes, and on the other side sometimes on the horizontal axis of this we are loading the samples, and on the vertical axis we have genes located, meaning they are the replicates sometimes. And suppose we are performing an experiment, we are comparing the normal cell with a cancer cell, and what we'll do is we are studying gene expression and we are trying to compare the gene expression in a normal cell with that of a cancer cell. What we'll do is we'll multiply this cell and culture this cell, and then we will isolate all mRNAs and then we synthesize cDNAs, and when we are synthesizing cDNAs we will use color dye. So we will have the different colored probe, meaning the samples, the cDNA coming from the two samples would have different colors. So one usually is red and the other is green. So what is happening is that some of the when we pass this through the microarray chip, what will happen is this cDNA will bind to its complementary DNA which is loaded on this glass slide. So some DNA which is coming from the samples would be retained on the glass slide and it will emit the color accordingly, meaning if no retained it will have black color, the one which was original. If the red color retained it will have red, if the green color retained it will show you the green color. So or if both are retained it will show you yellow color. So and this color intensity is analyzed. This is what we do in DNA microarray, and this color intensity is analyzed with the scanning machines and the data is normalized, and we try to understand the quantity by the pixels. The pixels are quantified and the data is normalized, and what we get as a result is the gene expression values. They are analyzed and they are clustered, meaning you get a heat map like this, and what you see in this heat map is you get the samples which are showing similar gene expression, meaning they are clustered, or you might also get the genes which are clustered during this expression. So we get huge data and we need to have the information about the samples, we need to have the information about the platform which is used, and we also need to have information about the expression. So this data is a unique type of data and we have to have special methods to analyze this data. So to summarize, what we were trying to understand in this lecture, that the gene expression data is is a different type of data, not normal sequences, and we have to have design special methods to organize this data and to understand this data. So I will close this section, but just I will summarize that microarray data in this nutshell is that data needs to be stored properly, like we have data before the experiment and we have a specialized data after the experiment, like scanned images or DNA expression matrix, that is the pixels, the relative expression levels expressed by the pixels. So I would just summarize quickly. What we learnt in this section is that we were talking about gene expression, though the gene expression is very trivial we study that in earlier classes also, but global gene expression makes it special, and we can take out several hidden facts when we study global gene expression, where we study hundreds of the genes together, and this throws a light on interaction and regulation of genes also. And we also studied ESTs and DNA microarrays, that they are yielding enormous amount of data. So thank you very much. We thank you for your listening, and in the next section we'll continue with the DNA array, with the gene expression databases, and we'll take the example of GEO there. Thank you.